We are now recording, great. So we can look at different fluorophores that have very broadly overlapping spectra. So this is an example now of the fluorescence spectrum from 9 different fluors. And despite the fact that they all overlap substantially, they can be distinguished by fluorescence spectral imaging. They can be distinguished by both the shape and the peak of the emission spectrum. So we do this with a relatively expensive microscope, and if we want to see more kinds of organisms than we have distinct fluorophores, we can also employ an approach that we developed, combinatorial labeling and spectral imaging, or CLASI-FISH. And the benefit of this is that if you can distinguish, for example, 8 fluorophores with your imaging setup, and then you use those 8 fluorophores in, for example, binary combinations, so that each different kind of microbe or each different kind of anything you want to distinguish is labeled with exactly 2 of these fluorophores, then in this case with 8 fluorophores, there'd be 28 unique binary combinations. So we can see larger numbers of kinds of organisms if we so wish. And just one more slide about the method. So this is an example from our earliest paper, almost 10 years ago now, about CLASI-FISH labeling, in this case of just as a test object proof of principle, a mixture of 28 different E. coli. They were all E. coli. We were labeling them with probes targeting ribosomal RNA, so we had to label them in 28 separate tubes and then put 2 different probes per tube to create these binary labels on these E. coli, and then we could make a mixture of these differently labeled cells, image them with the microscope and try to discriminate the different labels that we had put there. And then we counted the number of bacteria that went in bearing each label, and then the number that our imaging assay could record, and we got pretty good correlation between the two. So this is our proof of principle that you can in fact use spectral imaging with probes targeting ribosomal RNA to discriminate large numbers of kinds of organisms. We went as far in this proof of principle as using 16 fluorophores in binary combinations. That was 120 binary combinations. Then we decided that, yes, we could keep pushing the technology, but we were really more interested in looking at actual biology, right? So the question arises, how many different taxa do you really need to see to say something interesting about a microbial community or to learn how a community is structured? So that question actually led us into a fairly lengthy excursion in bioinformatics in looking at the sequence data from ribosomal RNA genes and trying to understand the data at high resolution and how much we could find out about what is actually in a community. And here I'm going to show now an example from the human oral microbiome. So the human mouth is a fantastic place to do microbial ecology because it has a number of different habitats within the same mouth and different taxa that live in those habitats. So we say here that most of the taxa are site specialists. So if you look at the bacteria that live on your teeth, they're for the most part entirely different, species from the bacteria that live on your tongue, which are entirely different from species from the bacteria that live on your gums, right? So this is a plot from a paper that we wrote last year showing, so that the size of these dots is it's related to the relative abundance of each of these kinds of bacteria in a sample from the mouth. And then these axes here are the ratio, here on the y-axis, it's the ratio of abundance in dental plaque to abundance on the tongue. And what you can see is there's a cluster of species that are 100-fold more abundant in plaque, at least 100-fold more abundant in plaque than on the tongue. Conversely, about the same, there are taxa that are very abundant on the tongue, less abundant in plaque. And then this x-axis here is showing the same kind of ratio, but for the gums, the keratinized gingiva. So you see a cluster of taxa that are 100-fold more abundant on the keratinized gingiva than they are in plaque or the tongue dorsum. So I think this is really pretty amazing because there is no barrier, obviously, inside a single mouth. Not really a significant barrier to dispersal, right? These microbes are all being shed into saliva. They're being transported around the mouth by saliva. They're all in the same salivary environment. They're all exposed to more or less the same immune system. And yet you find these very different communities at the different sites. Okay, so that's, first of all, just biologically extremely interesting, but it also, from just a technical standpoint of trying to image, it gives you a quite precise idea about which bacteria you need to be looking for if you're imaging a sample from one site in the mouth or another. So we know there are about roughly 500 species of bacteria that are resident in the mouth, but of those, there's only a subset of a few dozen that's really abundant at any particular site within the mouth. So that meant that we could develop a probe set for imaging that would target the key bacteria that are likely to be abundant in the particular sample we're looking at. We designed most of our probes at the genus level. In many cases, there are only one or two species in any particular site in the mouth within any given genus. So that gives us actually, we find it's a good balance of specificity to breadth as a general rule. So this is showing a probe set that I put together with probes for 9 different highly abundant genera within the mouth, each of them tagged with a different fluorophore, and then a probe for more or less all bacteria, the commonly used universal bacterial probe. And the diagram here is showing you where roughly these probes land on the ribosomal RNA. They happen all to land at different sites. That wouldn't have to be the case. In fact, it would almost be better if they were competing against each other, but that worked out. Okay, so when I applied this probe set to pure cultures first.
Quick question.
Yeah.
So you were talking about the combinatorial part of the probes Well, in this case, so if you go back to the previous slide, in this case it would be the blue bacteria and the pink, for example, or the green for Streptococcus. In this case, did you guys do combinatorial labeling, or this was just a simple, just like a one label?
Yeah, so this is simplified so that there's one label per taxon plus the universal probe. So in a really rudimentary way, it's combinatorial in that every bacterium is going to have 2 different probes on it, but one of them is just a universal probe. So this is essentially, we've simplified it down to single fluorophore per taxon. And we did that for reasons of especially when we were first doing this, in principle, if you have overlapping bacteria, in a densely populated biofilm, for example, any particular pixel can have light entering the pixel in your camera from more than one bacterial cell because of the depth of the biofilm and the overlapping, right? So we were worried that there would be a problem where you'd get ambiguous signals. You'd have, if you see 3 fluorophores, you wouldn't be able to determine, what kind of overlap was producing that signal. That problem might be a little more theoretical than actual. Anyway, this is where we started. So we're starting with a relatively simple labeling scheme right there. Okay, yeah. So then it looks real simple. Then when we apply these probes to pure culture, so this is just our proof of principle that when we apply this entire probe set to pure cultures and then do our imaging and linear unmixing, that we get the expected signal down the diagonal here, and then every cell is also hybridizing with the universal probe, and we don't see a significant off-target reaction. I should say that these columns have been normalized so that the positive cells here are bright enough to see. When you look at the universal probe channel where all the cells are labeled, so these in the universal probe channel here, they're all normalized to the same, they are all shown at the same experimental condition, and you can see in this channel that some of the cells are much brighter than others. So these guys down here are bright, these guys here are dim, and that is the kind of difference when you move from your proof of principle with E. coli to a real community where cells have different intensity for a variety of reasons. That starts to introduce technical problems, which was another reason that we kept the labeling scheme very simple in this case. Okay, so when you apply this probe set now to natural sample. This is a picture of human dental plaque that we sampled just with, toothpicks, the way you'd normally clean your teeth. We have to fix the sample to do this procedure, so everything, unfortunately, is not live but fixed. And then we drop it on the slide, apply the probe set, do our imaging, and we get this kind of an image, which right away you look at this and you see, okay, this is the benefit of being able to see spatial organization, right? Because you look at so you have this taxon Corynebacterium shown in this magenta color, this guy here, that is forming these filaments radiating out from a central core. Corynebacterium had always been there in the sequencing data, but had been largely ignored. Not that many people worked on it. People just didn't think about it very much. As soon as you look at this, you say, Corynebacterium, that's the foundational taxon. It's creating the spatial structure that the other taxa can then inhabit, right? That's an inference that we can make from the spatial organization here. Whereas the Streptococcus forms this shell around the outside. We thought that was really interesting because it implied that the Streptococcus was occupying this aerobic zone, about the outer 20 or so micrometers of the biofilm, and then apparently was modulating that environment to create an anoxic environment, the sort of internal ring here that contains Fusobacterium and Leptotrichia, which are generally thought of as anaerobes, or at least microaerophiles. So what we interpret from this spatial structural information is that the Corynebacterium is providing the spatial sort of organizing force for this biofilm, and then the Streptococcus is doing a lot of modulation of the biochemical environment. So that's 4 of my 9 probe set. Here's another 4 taxa that were in pretty high abundance in this biofilm. So you can see, again, the complexity starts to kind of layer on there. Capnocytophaga, shown here in red, is an organism that requires CO2, which we think is secreted by the Streptococcus. Several other taxa are participating in these little structures around the edge, which, by the way, are called corncobs, and I'll show you a close-up view of them in a second. And one interesting thing here is that amongst these taxa that's in this outer aerobic shell is Porphyromonas, which the Porphyromonas that everybody studies, Porphyromonas gingivalis, is a strict anaerobe. And yet the presence of this taxon in the outer shell suggests that it is a different Porphyromonas that's an aerobe. Actually, there's 2. In fact, there's these guys in the outer shell, and then there's some little rods that are in here. And it turns out in the sequencing data, there are 2 really understudied Porphyromonas taxa that are in rather high abundance. We are suggesting that this is some evidence that they may be aero tolerant. Okay, so these structures around the outside are called corncobs. And prior to this imaging, these corncobs had been described actually in the 1970s from electron micrographs of plaque. They were thought to be a 2-taxon structure consisting of this Corynebacterium, sort of the cob there, and then Streptococcus, particularly Streptococcus cristatus, as these kernels. We saw more complexity than that with regularity in dental plaque from different individuals. We saw this taxon Porphyromonas, some not very well characterized taxon of Porphyromonas, is also able to participate as the organism binding directly to that central filament. And then another taxon, Haemophilus or aggregatibacter, again, not very well characterized as yet, forming an added layer on the Streptococcus, but never, as far as we can see, on the Porphyromonas. At least whenever we see that aggregatibacter, it is touching a Streptococcus, and if it's touching Porphyromonas, that might be just coincidental. So we can see much more complex structure than we had previously thought existed. And actually delving a little more into that now. So all of that previously has been published. This is some unpublished work that we're writing up, looking more precisely at the species level, or at least closer to the species level, at these Streptococci in the corncobs. And what we see is, as expected, Streptococcus cristatus is involved in these corncob structures, but also an organism in the Strep mitis oralis infantis group, And these corn cobs can be either pure one of those taxon or a mixture of the taxa. So this is just an example of how far down you can start drilling just with imaging into how complex these communities are and how they're composed. Again, this is writing this up for publication now.
I have another quick question.
Yeah, please.
So I was wondering how reproducible are these structures? Like how many how many samples do you get, and do you see it in all of the samples or in most of them, or is it yeah, like how prevalent are these corn cob or just in general the hedgehog structures? Do you find it in all of your samples or yeah?
Yeah, so that's a very good question. So we have found the hedgehog structure. The reason it gets a little complicated because like, what exactly do you define as a hedgehog, right? So we looking in plaque, you see a lot of complexity. If I define hedgehog as it's got the Corynebacterium bush, it's got corn cobs around the outside, and it's got either Fusobacterium or Leptotrichia as that intermediate layer. We have seen that in, I would say, every individual that we've sampled more than once. But you can collect a sample from an individual that has none of that and instead has, a variety of other little cocci piled together. There are some samples that are almost entirely hedgehog structure, and there are others that have very little. So there is a lot of variability. Corn cobs, similarly, I would say pretty much every plaque sample that we've looked at has at least something that's corn cob. But again, sometimes in very high abundance, sometimes in much lower abundance. It's like sort of wading into the jungle and you're trying to characterize exactly what you see. Yeah, so yeah, it's complex.
Right. That makes sense. Okay.
Yeah, but it's a great question. I don't know.
Can I ask a similar follow-up question? I'm curious, so you're saying like within one person, sometimes one sample will look different than another. So do you think you see more variation across people still, or do you see kind of about the same level of variation when you look across people versus like within one person from one sample to another?
Yeah, that's another really good question. And it's a little bit easier to answer that question with sequencing, and I didn't throw these particular slides in there, but so from analysis of the sequencing data, we know that the community within a single person's mouth fluctuates over time substantially. The community from person to person, I should say, so from person to person, we all basically have the same species, certainly the same major species. I would say we basically all have them. But we differ from one another at the strain level. So the bacteria in my mouth will be the same species as the ones in your mouth, but we each have our different strains. And then the relative abundance of all these different taxa in your mouth fluctuates over time, over daily time scales, fluctuates quite a lot actually, but sort of fluctuates around a mean that is characteristic for you, right? So there are some people who have a whole lot of Neisseria, there are some people who have a whole lot of Porphyromonas on average, but it fluctuates over time. And one of the things we would really love to know is why does it fluctuate? What, is it waves of phage infection? Is it something the host is doing? What's going on that makes those fluctuations happen? What sets the boundaries on them? Yeah, there's a ton of really interesting questions there.
Cool, thank you.
Yeah, thanks. Okay. Oh yeah. Okay. So then this is my summary of plaque, and we published this image or this diagram in 2016. So this is our hypothesis, sort of put together from all those various observations of how plaque is structured, with this bush of Corynebacterium rooting on, we think, a pre-existing biofilm. So there are early colonizers that can come onto a clean enamel surface. We think that the Corynebacterium isn't doing that, but that it comes in about 24 hours into the sort of ecological succession. And then once Corynebacterium has established itself and made this bush, then it can be inhabited by all these other organisms, which we think are then doing things like converting, sugars into CO2 and lactate and acetate, taking oxygen, turning it into peroxide to try to kill their neighbors. All sorts of interesting interactions are going on that we'd love to know more about. So that's plaque. But when you take that same set of probes and apply it to stuff scraped off your tongue, you get this completely different picture. So this is something that we just published a few months ago. This is the biofilm that you see when you take, so let's say we started doing this more or less when my dentist handed me a tongue scraper and said, here, you should scrape your tongue. And it was this little ridged piece of plastic. I'm like, oh, great, we can take samples, right? So if you clean your tongue the way dentists tell you should, you scrape this sort of alarming quantity of material off your tongue. And then if we, again, fix it, drop it on a slide and apply probes, we see structures like this. And so in answer to the question, how often do you see a hedgehog? Yeah, good fraction of the time, but not always. These structures here we see in every sample from every person that we have ever sampled the tongue of, right? So these are found regularly, routinely. There's a core here of host epithelial tissue. What we think this is actually is your tongue is coated with papillae. There are fungiform papillae that contain the taste buds. There are 10 times more these filiform papillae that have these little hairs projecting off of them that are covered with bacteria. So we think that this is a hair projecting off a filiform papilla with all these bacteria. And obviously growing in also a rather organized, structured way, but completely different from the structures that you see in dental plaque. So for example, here I'm talking a long time about this one slide, but it's pretty cool. So actinomyces, which is generally anaerobic, right? It tends to be right around the base near this sort of host epithelial tissue. And then Rothia here shown in the sort of cyan color tends to form sort of large patches, and where the actinomyces tends to be wider at the base and getting thinner as it goes out to some degree, then the Rothia tends to kind of make larger patches more in the aerobic zone. And I point out both of those taxa and also Vianella here because they are taxa that are capable of reducing nitrate to nitrite, which is one of the functions that is hypothesized for the oral microbiome in general and the tongue microbiome in particular, is to carry out for us this step in metabolism that our bodies don't carry out very well, which is conversion of nitrate to nitrite, which your body can then take to NO, which dilates blood vessels and has all sorts of effects on the body. So that's a hypothesis for why we might be at least tolerating the presence of these bacteria in high density in our mouths. And another interesting question, well, okay, so I should say, so we see these consortia in every person that we've ever sampled. They have variable form. What we can quantify in them is the relative abundance of the taxa that we can see, right? So you can faintly see that there are these sort of dark patches. We haven't been able to stain everything in these consortia, but the 5 or 6 different types that we have stained here make up a high fraction of the cells and in rather variable abundance from consortium to consortium. And then also here, this is from person to person, right? So these are 20 consortia from one person, 20 consortia from the next person. And you can see that, for example, some people have a higher proportion of these Neisseria, I think, that's labeled in yellow. Others have rather few, so people have different reasonably stable differences in proportions of taxa, but compared to the fluctuations in relative abundance of the different taxa, different people can look at this data. Some people will say, wow, that's really high conservation. And other people say, wow, look at how rapidly it changes. So it depends where you're focused, right? But we see very consistent consortia overall and really surprising consistency in the taxonomic composition of them, I think, from person to person and consortium to consortium.
Jessica?
Yeah.
I was wondering about these structures. I mean, these are from, you're saying, tongue scrapings, basically. So I imagine the scraping kind of disrupts the overall organization, kind of vertical organization on the tongue. So these are kind of like little nanostructures within the entire biofilm that are just quite stable and not disrupted by the scraping. Is that the hypothesis?
Our hypothesis, let's see if I have a better picture. Yeah, so our hypothesis is that this pore here is a hair kind of sticking up from the papillae, sticking out into the saliva, and that this is the biofilm that has formed around it. And actually, there are electron micrographs that show basically globs of bacteria big round globs of bacteria sticking out toward the saliva. So what I think is going on here is that this is the consortium more or less as it is in life. And actually, so for example, you see in this image somewhat this little crust of Streptococcus. We often see at the edge of the consortium just one or two cells thick, a little row of Streptococcus. And that signifies to me that that is actually the surface of this biofilm. So it's not so much a single mass of confluent biofilm as a whole bunch of little pieces of biofilm sticking out into your saliva. So, and we think when we scrape them off that what's actually happening is that the epithelial cells, which are preparing to be shed anyway, are just coming off a little bit sooner than they might have otherwise. So we're dislodging the whole structure by itself. That's at least what we think. Next step in that, of course, would be to get a biopsy of a tongue or something and see if we can prove it.
Got it. Okay, thank you.
Yeah, sure. So let's see. Oh yeah, and then the other thing that we see then when we look at a scrape from the tongue, from any one grape, we can see these consortia in what appear to be different stages of development. So again, this is all from a single time point. Yeah, and I should point out the scale bar here is 30 microns. So each little dot here is a bacterial cell. Here it's 5 microns. Yeah, so each dot is a single bacterium. And yeah, so at a single time point on your tongue, there will be these structures in these different, what we think are different phases of development, right? So a relatively thinly colonized little hair here, and then thicker colonization and thicker colonization yet. And then what would limit the thickness of the biofilm, then our idea about this is that what limits the thickness of the biofilm is the rate of shedding of the underlying substrate, right? So that presents an interesting idea for how the host could control certainly the thickness and potentially also the overall structure of the biofilm simply by controlling the rate of shedding in the environment. So everything that's in your mouth is in an environment of flow, right? Salivary flow is continually going on. We swallow between half a liter and a liter of saliva per day. So there's this continual flow through the mouth, which means that any bacterium that wants to be in the mouth has to hang on, or else it's going to end up in the stomach, right? It doesn't want to be in the stomach. So everything in the mouth is adherent, and the length of time that that substrate it's adhered to is present in the mouth is going to determine the length of time that adhered organism can be there, right? So that it provides, so the rate of flow as well as immune system and the composition of saliva and other factors is one means for the host to be controlling and shaping the microbial community. That idea is consistent with what we see when we look at buccal epithelial cells, such as cheek cells that we scrape off our cheeks. This is just one picture I have of this that I'm showing, these sort of faint gray objects, each of those is a buccal epithelial cell, and you see that they're rather thinly colonized by usually more or less just a monolayer of bacteria, and often a rather simple layer that's rich in Streptococcus, and in this case, Gamella. And that's consistent with the fact that the rate of shedding of buccal epithelial cells has been calculated at approximately renewed every even 3 hours or so, right? So the substrate there is not present in the mouth for long enough to develop this thick biofilm. By the way, I think I forgot to mention that all of these images I've shown so far, these are confocal images of three-dimensional structures. So this is basically an optical section through a much larger 3D thing. Whereas for buccal epithelial cells, what you're imaging is much flatter in the first place. So contrast that, what's going on in the mouth, with what goes on in the gut, so the gut is also characterized by flow, right? There's peristalsis carrying material through the gut, but its flow within an enclosed system. So a lot of interest on the gut is focused on the proximal colon, because that's where a lot of the microbial action is going on. The residence time of a microbe in the proximal colon is something on the order of like 12 hours. In the proximal colon of the mouse, 12 to 24, somewhere in there. So this is a picture of a cross section of mouse gut, mouse colon, and I've stained this so that you can see the mucus. So mucus is stained both yellow, actually red and green. The overlay is where the two mucus stains are. So you see these goblet cells secreting this layer of mucus that is separating all this business going on in the center of the gut from the host tissue. This business that you see here in the center of the gut is just from autofluorescence. These are food particles in the mouse chow, right? So it's plant matter in mouse chow. And also you can see the mucus very brightly staining in this layer, but also distributed throughout the gut. And so any microbe that is in this environment here, it may be adhered to a food particle, to the mucus, it may be trying to adhere to substances so as to localize itself into its favorable metabolic environment. But whether or not it's adhered, it's going to remain in the gut environment until the passage of all the contents of the gut out of that environment some hours later, right? So there is some requirement perhaps for adherence, but it's not nearly as comprehensive, as essential as it is for a microbe in the mouth. And perhaps for that reason, if you zoom in now, so now I've zoomed into the wall of the gut, and now I'm staining for bacteria. So here's the host tissue along the side. Now I have not stained for mucus, so this gap in here where you see nothing, presumably that's the mucus layer. And then here I've labeled 5 different kinds of bacteria with different fluorophores again, and in such a way that you can distinguish the Bacteroides in these sort of cool colors. And the Bacteroides tend to like to be living right in that densest mucus layer. You can distinguish them from some of the other bacteria in the center of the gut. But so the most striking thing about this picture to me, as also food particles are now labeled bright yellow by autofluorescence. The most striking thing about this really is the lack of much structure, right? There's these beautiful microbes, but they're very, very intermixed with one another. And that really surprised us when we first took these images. So we imaged the gut expecting really to find quite a bit of structure and quite a bit of difference between the community very close to the wall of the gut, right there in the mucus layer, and then the community in the center. And what we saw instead was really a very high level of mixing, which surprised us because, purely by the process of cell division, bacteria will form little clusters, little single taxon clusters in which they are right next to their sisters. And what this shows us is that the rate of mixing, the rate at which those clusters can be disrupted, is more or less comparable to the rate of cell division. Right? There's really a surprising, to me, degree of mixing in that community. This extended even to in the crypts here between the villi. We do see some bacteria kind of invading into the crypts. So these crypts are presumably full of mucus, which is exuding outward, and we do see some bacteria in there. There's an idea out there that those bacteria might be actually forming little microcolonies in the mucus layer or on the epithelial cells, and from there they might be seeding the gut. At least in this somewhat artificial system in which we were looking. I'll say a moment about the artificial system is this is a notobiotic mouse, so it's a germ-free mouse that has been colonized then with bacteria that are characteristic of the human gut. So it's an artificially simplified system, and you also have microbes that are not adapted to this exact host, right? So with those caveats, what we see is that the bacteria that do get in here into the crypts are also rather mixed. There are individual cells next to bacteria of other types and not next to their sisters. So this gives the appearance that these microbes have recently invaded the crypt from this more mixed mixed environment of the lumen of the gut. Okay, so, and let's see, actually, one more thing to say about this. We can distinguish some spatial structure, so it took a lot of sort of looking and thinking to figure out what, if anything, we could say. And the one thing that I could say with confidence about this data is really that the bacteria that I've labeled here in blue, the Spectral cellulose-lyticus, which was the most abundant of these bacteria in this system, in the areas where they were the most dense, this Ruminococcus torcus was rare, and vice versa. And in particular, right at the edge of the mucosa, you had a tendency for the Bacteroides cellulose-lyticus to be dense, and a tendency for this Ruminococcus to be excluded. But as you can see, the error bars are rather wide, right? So quantitative analysis can be done. There is not total absolute mixing, but there is a great deal of mixing going on.
Sorry
Yeah.
to interrupt you.
Yeah,
So in the gut, we are seeing, like in these mice, I missed the part that if you infected these mice with these bacterial communities as a 1-to-1 ratio, or do they kind of, there is something that some of the species or taxa is taking over? So to me, it's like, the mixing is happening, but is it like we have one species that is taking over and then the rest are there as a pure kind of like less abundance? Is it something you always see, or is it based on the inoculation at the beginning and then it's just
also really interesting. Yeah, good question, good point. Let me see if I can say this based on any of this. Yeah, so these were, it was a 15 taxon community introduced at what so this is actually with Jeff Gordon's group at Washington University. Nathan McNulty, working with Jeff Gordon, actually did the mouse end of the work, and he tried to introduce the bacteria in equal numbers. And what happened was that this Bacteroides cellulose-lyticus, that made up like almost half the total, right? That one thrived in this community in a way that a bunch of others didn't. Of the 15, I think 6 of them were rare enough that you could easily detect them by sequencing, but not so much by imaging, because they were, you'd have at most 1 cell per field of view, probably less. So yeah, there was definitely differential success. But, and then I guess I can say I don't, and I should also say this was more or less just a pilot experiment that ended up, just working out how to do the imaging ended up being complex enough that we don't have a very high N to tell you what happens if we did this 20 times, right? I don't know. But what we do know is that even on this sort of micron scale, the Bacteroides do have a tendency to be abundant in the really dense clusters of mucus. I think that the Clostridia were abundant on the food particles, but I could never prove that. The statistics weren't good enough to really show it. So I think that there are differences in how successful the different microbes are in the microhabitats in the gut.
Yeah.
But the differences were modest enough and the mixing high enough that it was very difficult to prove.
Yeah.
That would actually be an interesting field for.
Yeah, exactly. I was thinking it opens a lot of doors to kind of a study between like the competition between different bacteria in their environment. But thank you. Yes.
Yeah, exactly. Yeah. Fantastic. Yeah, there's so much to do.
Yeah.
Yeah. Mice are a little bit complicated. It would be nice if the simple system were a little bit simpler, but hey, mice are good. Okay, so.
A quick question.
Yeah.
How long were these mice colonized with this community before imaging?
How long were they colonized?
Yeah, before imaging.
Yeah, I'm trying to remember. I think it was at least a week, but I think it might not have been much more than a week. So like, 6 or 7 days to establish the community, and then killed mice and do the imaging. So, and again, yeah. So what would happen if it were longer? Maybe it would change. Yeah, there's all sorts of possibilities.
Thank you.
Yeah, we collected some fecal pellets as well. One could also analyze what's going on in the fecal pellets, where it's much easier to do a time series. There's not this sort of hard endpoint to the study. But what people are largely interested in is what's going on right there at the mucosa. So we had to use actual tissue samples. And I should say also that these samples were, we took the tube of the gut and embedded it in acrylic, basically, a methacrylate resin. And once in that methacrylate resin, everything is just stuck and not moving. Because another thing that we worried about is that we would just, in the handling of the samples, disrupt the organization, which is why we just took the tube of gut and immediately stuck it in plastic and then sectioned it from the plastic.
Jessica?
Hey, yeah.
Thinking about what you just said about fecal pellets, it does occur to me that this would be a great way to validate how the fecal microbiota actually compares with the gut microbiome, because, I mean, especially for human studies, it's always stool sequencing that is kind of used as a proxy for gut microbiome, and we don't really know how accurate that is, and obviously it's not generally feasible to go straight into the gut and sample there, especially not over time. So I was wondering if there had been any kind of formal comparison of fecal microbiome versus gut microbiome, and if they're actually at all similar or close to similar.
Yeah, people have done that again by sequencing, compared species to what's in the lumen of the gut and what's in the mucosa of the gut in various model systems. I'm not sure. In principle, you might be able to do it in humans also with biopsies. So in my sort of view of it, which is not really the majority view, is that that lumen. So the big argument really is our lumen and mucosa. What's in the middle of the lumen? Let's see if I can get a better picture here. But yeah, okay. So what's in the lumen here in the middle versus what's here right at the edge of the mucosa? Are those the same community, or is there a community that resides here at the edge of the mucosa? I think partly based on imaging and partly based just sort of on first principles, we know that this mucus is shed rapidly and constantly basically into the lumen, and we know that the epithelial cells are shed. I think it's really basically the same community that just gets rather thoroughly mixed. But there's certainly some findings in disease that you can get a different community right here at the edge of the mucosa, and even particular strains. There'll be Bacteroides fragilis. There might be some strains kind of hanging out in the lumen, and other strains right at the mucosa in disease. But that's, in my opinion, where there's been a disturbance in this continual shedding of mucus and epithelial cells. So anyway, so there's dispute on that point. I think that the fecal samples are a pretty good sample of what's going on in here, except that during the period of time between when they leave the proximal colon and make their way all the way out to become feces, shifts in composition can happen during that period, which might be a day. Anyway, that's my personal take on it. Anyway, so my personal take is that feces are a pretty good indicator actually of what's going on in the gut, but there's certainly argument about it.
Okay, well, that's good to know. Yeah. Sorry, there's probably a lot of literature on this already. I haven't really been up on it, but it's good to hear that there's continuity between those data sets.
Yeah, I think so. Again, mine might be somewhat the minority opinion, but that's my opinion. Okay, yeah. So anyway, so that's what I can tell you about the mammalian microbiome, and then I'll just mention that. So these same methods can be used to study your favorite microbial community, whatever it might be. And I have 2 slides here of examples. So this is a study that we published actually just this year on using CLASI-FISH to investigate microbes on marine plastic debris. The plastosphere, ultimately would like to understand what is the fate of plastic in the marine environment, and in particular, how is it digested, and are there microbes that are particularly good at, specifically digesting the plastic in the marine environment. So this was step one of, can we image these microbes at all, and then ultimately then figure out who's sitting right there in the little pits that we can see on the plastic, and can we figure out how to encourage that kind of process. And so what you see here are some images from plastic debris that was just incubated, actually just like off the dock in Woods Hole for 1 week, 2 weeks, 5 weeks. There are some diatoms here with little individual bacteria attached, and then some other sort of micro eukaryotes of some kind. So yeah, so anyway, it works in the marine environment on plastic debris. And then here is CLASI-FISH on the kelp, the surface of kelp, Nereocystis. So here is a sort of head-on view where we're labeling several different types of bacteria, and you can see some spatial structure in that some of them grow in these big clusters. Granulococcus, which is a common symbiont of kelp, grows in larger clusters and then is surrounded by alpha proteobacteria and other Bacteroidetes. And then this is a cross section. So again, this is the kelp blade embedded in plastic resin so that we can make a cross section. And you can see it on both surfaces. Here's the surface here with this microbial community, and then another surface on the other side. And you see the little filaments projecting out into the water column, and then also some of the bacteria sort of invading into the tissue itself. So we're interested in trying to understand what role these bacteria might be playing in nutrient transformations and in the biology of the kelp itself, and to what degree these are bacteria attaching to any available substrate versus to what degree they are really symbionts of the macroalgae of the kelp. And I guess I should say about that we do see a rather consistent community on macroalgae on kelp that's different from the community you might find on a rock that's next to it in the environment. So these methods are broadly applicable. And then just to sort of summarize, I'll say that we think looking at spatial organization of complex communities at these micron scales, the scales at which the bacteria themselves really live and interact, gives you information about how growth of certain taxa might be creating conditions in which other taxa can thrive, serving as attachment substrates or modulating oxygen concentration, or they have other roles. And they let us develop hypotheses, at least, about how the host might be establishing key parameters, such as the properties of the epithelium, properties of the mucins, the rate of shedding of the epithelium, and rates of flow that influence the composition of the community and its structure and its dynamics. And I should thank all the people who are involved in this research, names listed here. In particular, I'll just mention Gary Borisi, who really started this project as the director of the Marine Biological Laboratory some 15 years ago, and I was right in there with him originally as a postdoc, actually, and I've just continued working with him ever since. It's been a very productive and enjoyable collaboration. That's great. And now I will stop sharing my screen so I talk too long. And
awesome.
Let's talk about it.
Yeah, thank you so much, Jessica. This was fantastic. Those are lovely pictures.
Thank you.
So yeah, I want to open the floor for any questions that anybody may have.
Jessica, this is Marvin. I had a question. I had to step away a couple of times. I hope I didn't miss it, but I was thinking with this technology, so you can get some assessment of what bugs are there, how they're spatially organized. What else can you get out of it? Can you get out anything about growth rate, metabolic activity, anything like that, do you think?
Yeah, so in order to do what we are currently doing, we have to fix everything.
Yeah.
And that makes it much harder to do all those things, so where we really want to go is to completely change our technical methods so that we can do this on live communities, to get at growth rate and to be able to measure, aspects of metabolism. That would be just fantastic. And we're trying to push it that way with live cell imaging.
Yeah.
But that's still in its infancy. Yeah.
People have used like P&A fish and others to get at a rough, assessment of the number of ribosomes. Does fluorescence intensity sort of equate to that for you, or?
Yeah, people treat fluorescence intensity as if it equates to ribosome content, and it in that it's a probably well, in some organisms yes, in some organisms no. Or rather, it's a pretty good measure of ribosome content, but whether that tells you anything about activity, that's the question. So E. coli will change its number of ribosomes constantly depending on what it's doing, whereas there are other organisms that have a rock solid number of ribosomes, and they just, no matter what's going on, they don't change that. So yeah, that's of moderate utility. If you know the organism, which kind your organism is, then that would be useful. Yeah.
Do you see a big difference in if you look at a single, let's say, taxa in your images, and you look at the fluorescence intensity, does it vary widely, or is it pretty narrow spectrum of intensity that you see across in a community?
That also actually is different from taxon to taxon. So there are taxa that are really bright and taxa that are really dim, and that's one kind of problem. But we can address that problem by using different fluorophores for the different taxa, making sure we apply different lasers, so you can separate them out, you can pull them out. What's harder is there are also some, even when we see this in pure cultures, some pure cultures, every cell has about the same fluorescence intensity. Other pure cultures, there's an order of magnitude difference.
Oh wow.
Or even more. And then that basically means you don't see the ones that are dim, right? So that's another kind of problem. And actually we're trying to work on that, even just using just light microscopy, it's flat out light microscopy, to be able to recognize the different kinds of cells so that we can, even identify cells by morphology. Now, the whole point of doing in situ hybridization is that morphology doesn't help that much with bacteria, except that maybe once you start having information about, how many taxa are there, we could get some information that way. So
yeah, it seems like once you start learning enough about where things are and what types should be where, you might be able to do that, right?
Yeah, exactly. You can sort of put the different kinds of information together and kind of work it out. So yeah, so it's never easy, right? But you can get so much amazing information that way that it's worth trying.
Well, thanks a lot. That was awesome.
Oh, thank you.
I had a question. It's James Kenny.
Hi.
Have you considered trying to make a probe for bacteriophage? Obviously I know they don't have ribosomes. You have, there's a universal system. There's nothing like that exists in phage, as far as I'm aware. But you could sequence the local phage environment, build a probe specifically for them, and ask. I haven't seen anyone do this sort of work where it's looking exactly where the phage are interacting with the populations in this sort of scale.
Yeah.
In my mind, it just requires knowing the sequence of the phage and picking a sensible target.
Yeah, exactly.
Or am I missing something? Okay.
No. So this has been on our list to try for quite some time, and we just haven't really found the person with the time and the inclination. What we think you would do is, as you say, figure out what phage you're targeting, and then instead of using ribosomal RNA where we have the natural amplification, you just have to use more probes. Or even back when I was back in my PhD, I would label 40 kb of DNA just with nick translation, right? So you can do that. When it's in the phage head, it might be hard to get the probes in there, and there might not be room. It might be too tightly packed. But if it were if it were actually in the bacterial genome, or if you had a prophage, then you could probably see it. So yeah,
that's even when it's injected. I don't know what kinetical wise. Once the phage DNA is injected, you maybe have 10 minutes. Is that long enough before? Is that long enough time window to actually get you can see active infections of the bacteria from the phage genome?
That would be cool. Yeah, you just have to fix it during that 10-minute window.
So we have collaborators of ours are working on phage therapy in CF, and we have the potential. We know the phage they're putting in. We know the bacteria they're targeting. So it seems like the system could be tested in that way. And we have, obviously Marvin has his SCFM2 media, so we have a semi-structured environment which replicates the actual lung as well. So it's worth thinking about, right?
Yeah, sounds like fun.
I think we just have 1 minute, which I would like to thank you for joining us today, and thank all of our attendees. And again, I invite you, if you would like, and if your group would like to join us for next week's, we would be delighted to have you all. But let's once more thank Jessica for joining us. Thank you.
Great. Thank you. It was
Have
a
a
pleasure.
great week.
Yeah,
Thank
you
you.
too.
Bye.
Thank you. Bye, everybody. Thanks for coming.
Bye.
Yeah.